Okay, good morning. Happy Friday. Happy rainy Friday. I hope all of you are staying dry wherever you are. Uh, Today, we're going to talk about the very important concept of the genetic code and how the genetic code forms the basis of how our cells know what to do and how our cells know what instructions to follow. We started talking about PCR last time as a way of, in the laboratory, artificially amplifying DNA in order to be able to better study certain sequences that we might be interested in. For example, in a particular organism, if you want to look at a particular gene, to be able to maybe look for things like mutations, you might use PCR. So we discussed the steps of PCR last time. So I wanted to follow up quickly by just going over a couple things about how we practically make use of the PCR experiment in the lab. So to start off with, remember that PCR is this process by which you are amplifying a particular sequence that you are interested in from something, let's say a sample of tissue. Let's say you get a sample of tissue and you want to identify, for example, what organism this is, what species this is. Those of you doing the 275 lab, I believe you'll be doing this. Right? So keep in mind that you get a sample of tissue, and that sample of tissue would have the entire genome of the organism, most likely arranged across many different chromosomes or individual DNA molecules that make up that organism's genome. Let's say the way you want to analyze or identify what the organism is that you know there's a particular gene in there that you are familiar with the sequence of that gene, and you know how it varies from one species of something like a fly, for example, how it varies from that species to another closely related species that might visually be very hard to identify if you were just to look at two of these flies. But you know that the genetic sequence of a particular gene is slightly different, and it's been cataloged so that if you can figure out what the genetic sequence is of this little tissue sample that you take from one of the flies, you might be able to compare it to a known database and figure out what species of fly it is. So you'd start off by picking what genetic sequence in the genome you're interested in amplifying with PCR. Remember, the purpose of doing PCR is normally you have just a little bit of material to start with, right? So it's difficult to get a very clear read or a good, strong signal from that. In the previous lecture, I used this kind of analogy of if you have a boombox, portable radio sitting in the middle of a big campus by itself, and it's playing a song, right? If it's just that tiny little boombox in a huge campus and you're several hundred yards away, you might be like, oh, I cannot recognize that song. I think I know what that song is, but I can't really hear the lyrics very clearly. Right? But if you amplified that, so I think in my slide, I made the boombox humongous, but really what we're saying is you're not taking a small piece of DNA and making it humongous. What you're doing is you're copying it perfectly many, many, many times. So essentially, instead of having one little boombox sitting in the middle of campus, you have 1 million of those identical boomboxes all playing the same song at the same time, right? Then you can really hear the signal clearly and make out all the details like the words, the instruments, whatever, right? So that's what we're doing with PCR. And so when I say signal, what I simply mean is when it comes time to try to read the sequence of this fragment of DNA that you're amplifying, right? The signal is much stronger and clearer and more reliable if you have many perfect copies of this DNA fragment that you're reading at the same time. So what you would do is you would say, well, I already know what this gene is because I've chosen that, right, as the gene I want to compare and I want to sequence it. So that means that somewhere in the genome is the target sequence. Now, keep in mind that even though the cartoon is just showing this as this little blue fragment of double-stranded DNA, this is really just part of a much longer strand, right? This is just one part of one chromosome that's maybe one of several chromosomes that are all in the sample of the cellular tissue that you got, right, that you extract the DNA from. Okay? Now, keep in mind, the DNA that you've extracted from, let's say, a sample might not be very stable, right? A big, long molecule of DNA might just start breaking down once you actually just have it in a test tube. But hopefully, the quality of the DNA is enough that this particular sequence is still intact. Because you know the sequence, because you're the one who chose it as the one that you want to amplify, you can design primers. Right? Because remember that during DNA replication in a test tube, if we're doing PCR, right, you're going to have 3 main steps that you go through. One is heating the DNA up to separate the 2 strands. One is cooling it down enough so that you can have these little DNA primers that you've designed attach. And each of these little red primers, there is a 5' end and a 3' end, and they will be complementary to the sequence that you're interested in. So one primer will amplify this template strand from left to right. The other primer is designed somewhere else, maybe at the other end of the gene that you're interested in amplifying. Of course, this little red primer, it's also 5' to 3', right? But because this is the other template strand, this blue strand itself is in the opposite orientation as this top template strand, right? Because remember, they're anti-parallel. You need primers because the third step of PCR is then you heat it up so that the Taq DNA polymerase, right, the big protein complex that can actually recognize the 3' end of a primer and start adding these DNTPs from all the DNTPs that are floating around in your test tube, right? It starts putting the correct one, next one, next one, next one. And so that your red fragment here, your copy strand, grows in a 5' to 3' direction. You can see here, as the DNA polymerase is reading along this, right, even though it reaches the end of what you're interested in reading, there's nothing to stop it from continuously reading because it's just copying the template. But at some point, it's just going to kind of trickle off, right? Because the DNA polymerase is just going to fall off, right? Likewise, on this one here, as it's extending from right to left, you're going to have some extra stuff going off along the rest of the chromosome, right? But the part you really care about is the middle part, because that's the part you decided you wanted to sequence, and that's how you made these 2 primers. So those are the 3 steps of PCR to make one cycle in, let's say, the PCR machine, right? So at the end of that one cycle of heating, cooling, and raising the temperature a little bit, you've encouraged then the DNA to come apart, the primers to anneal, right, or attach, and then you've encouraged the Taq DNA polymerase to start extending them. Right? At that point, you would then have 2 double-stranded DNA molecules, right? That's the end of one cycle. So then you go back to the start, you heat up both of those again so they separate, and et cetera, et cetera. So here we have 3 cycles of the PCR program, and it ends up starting from 1 molecule, you end up with 8 molecules. Notice that starting from the second cycle, right, these red fragments serve as the template for the next round. So as a result, over time, the little extra bits that are included for example, in the first round here, right, you got a little extra bit. It's not going to really be amplified in subsequent rounds because the primer, right, from here, if it anneals here and reads from left to right, there's nothing else to read as a template over here, so it would just stop, right? Likewise, the other primer that goes from right to left, when it anneals, right, it would anneal over here, so it would be ignoring the little blue stuff because it starts here and goes from right to left. So in the finished product, after not just 8 cycles, or sorry, not after just 3 cycles, but let's say you go for 35 cycles or more, right? Exponentially, the number of molecules go up, but you have an overabundance of specifically just those red fragments in the end, right? Millions and millions of those that are made, right? So how do we visualize these to see what we've made? we visualize these on a gel. So typically we use an agarose gel. So agarose is a long polysaccharide, long chain of typically galactose sugars that's extracted from seaweed. Right? So another great example of this macromolecule, a long chain carbohydrate that's used in the seaweed. I think it's actually a structural molecule that's used to give the seaweed, strength. Right? So if you have purified agarose, it's these long strands of these sugars. It's kind of like a mesh, right? Or a net. And if you imagine, if you go ahead and you pour a gel, it's all these fibers in this big meshwork, and then you allow it to cool down and harden, right? Because then they form this network, a three-dimensional network. So you have little spaces and big spaces between them, in which if you then had molecules trying to move through them, right, big spaces and little spaces, depending upon the size of the molecule that you're trying to get to move through this mesh, right, big molecules would get stuck, smaller molecules would kind of make their way through the little spaces between the polysaccharide chains. So that's what we do. We pour a gel of agarose, let it harden. We have little wells cut out in them, in which we can load our DNA samples that came from our PCR. Right, so after PCR, you go ahead and you load some of your PCR product that comes out of the PCR machine into these wells. And then you apply an electrical current. Remember, DNA is negatively charged because it has those phosphate groups hanging off at the 5' end that have a negative charge. Right? So as you apply a current, the DNA will want to be drawn from the top here, right, towards the bottom, because this is where the positive electrode of your power supply is, right? The negative one is up here. So the current, right, is flowing in a way that the DNA, the negatively charged DNA, is drawn through the meshwork of the agarose. And long strands of DNA, you can imagine, get caught up in the mesh. They don't move as fast, right? Small pieces of DNA, they kind of wiggle their way through the gel, and they would migrate further. And importantly, all DNA fragments that are the same size would migrate at the same rate. So then you visually check your results. After you run this for some time, maybe half an hour, maybe an hour, and your samples have been drawn through, you can then stain the DNA using, let's say, some sort of a special dye that actually binds to the DNA molecules and then allows you to then see it when you shine something like UV light on it. Right. Notice some of these lanes, you would have very bright, clear, single bands. Right? Other lanes, you would have multiple bands from, let's say, a PCR product. Why would that be? Well, it might be that these primers at the top here that you designed to specifically amplify one part of the genome that you chose, right? Maybe in reality, the sequence of these is kind of close enough to some other part of the genome as well, right? So you might actually not only be amplifying one part to make these red bars that you see at the top here, but maybe some other part is also getting amplified, right? And those would be a different sized band because maybe these two primers don't sit at exactly the same distance from each other on that other part of the genome, right? Maybe one sits over here, one sits over here, you get a bigger band, right? But those bands hopefully would not be nearly as bright as the one that you meant to amplify, because that should be the 100% perfect match of where these primers go. Right? You also notice sometimes instead of bands, you get a bright smear. So what's that about, right? One way to think about this is once you have this information and you're looking at it, right, how do you make sense of the sizes of these different bands? Because if you run the gel for 15 minutes, right, maybe the bands won't separate that much from the top of the well where you loaded them. But if you run it for an hour, then that's plenty of time for the bands to separate. So how would you actually make sense of what you're seeing here? You would have to have some sort of a legend or some sort of a key in here to refer to where you know where the different sizes of DNA bands should be. Right? So that would be a special sample that is pre-made that would have different PCR bands already mixed in sort of as a mixture. And each of these bands is a very specific size. So if you run those on the gel, you know that wherever these end up, whether they're still bunched together at the top, if you only run the gel for 15 minutes, or if they're really spread apart, if you run it for an hour, wherever those bands are, that's your key that you can line up where your bands are in your samples and know the size. So you can think about this using this toy, right? So this is actually, this is like one of the earliest toys I had when I was a kid, right? So it's basically just this thing, this little thing where it's made up of different size beads. Oh, that's what a sound, right? And you shake it around, it's a baby rattle toy. Okay. But this helps us illustrate the concept of how things separate by size in a gel to expected points within the gel, expected sizes. So here, let's say you have this special DNA ladder that you use as your key. So the special DNA ladder is a reagent that's already a pre-made mix of high concentrations of DNA fragments of predetermined size and predetermined number of molecules, right? So not only can you use this to tell the size of your PCR product, but you can compare the relative intensity of the brightness of the bands, right, with this DNA ladder to actually estimate how much DNA you made. So these little plastic balls of different sizes, they're all loaded up at the top of the well in this gel, right? And as you start running it, the different size DNA fragments in this DNA ladder, right, they begin to migrate at different rates depending upon their size. So at the end, when you stop your gel and you go ahead and you look at this under UV light, the different sized bands would neatly be at different spots in there. And this is also an important thing, right? The band that you see represents many molecules that are at that particular spot in the gel. When I was an undergrad, I got confused about this. I thought like when you see the bands that's actually like the DNA molecule from like right to left in the lane. I know in hindsight that sounds really, I feel kind of dumb admitting that. But I shouldn't say it's dumb, because if some of you also have that misconception, right, let's just clear that up now. In reality, just like these little balls in the toy, right, the size of the ball, like this yellow ball, does not stretch from the left side to the right side of the lane. It's just you have a whole bunch of them in there, and together it makes a band, right, just like you see on the toy here, right? So that's what it looks like. So a typical result would be you would have, for example, 4 sized plastic balls in this toy. You end up with 4 bands on the gel where these different sizes would be. Then you have, at the same time, of course, you're running in the second well your PCR product, right? So it ends up over here. Not only then can you estimate the size of your PCR product, because then you just look at the little product literature that comes with the DNA ladder that you used, right? It tells you this band is maybe like 500,000 base pairs. This one maybe is 700,000 base pairs, right? So you estimate, oh, my PCR product is about maybe 550,000 base pairs in size, right? And you're like, yeah, because originally when I designed the primers, that's how I designed them, because the region I was trying to amplify is 550 kb, or kilobases, right? And you're like, yay, that's my product there, right? What about the smear? This smear is what you would see if you took some of your original genomic DNA that you had collected from your tissue, right, that you used as the basis of your PCR, but instead of doing PCR, you just went ahead and loaded that in one well to look and see what it looks like. So not surprisingly, right, that's going to be a whole hodgepodge of different sized fragments of DNA just from the whole genome that has kind of started breaking down randomly just because it's in a test tube. So you get a smear, right? An infinite, well, not infinite, but many, many different sized bands that appear as a smear. Okay. Why then do you not see the genomic DNA smear in your PCR product? Well, because after all, didn't you start with this? So shouldn't this be in here? It's because the relative amount of DNA that ends up being made, right, you've greatly amplified just this fragment here. So if you were to normalize the total amount of DNA that you're loading in sample well 2 and the total amount of DNA that you're loading in sample well 3, right, so much more of the DNA in your PCR product is your band. So once you run it out, you don't even see the smear because it's so faint in the background relative to this huge amount of the fragment that you made. Right? So genomic DNA would be a smeary mix of randomly sized fragments. The larger pieces of DNA can hold more dye molecules on them. So that's why they glow brighter at the top. Right? That's why the smear looks brighter at the top. And the smaller DNA molecules, right, wouldn't hold as much dye in them. All right. So successful PCR leads to immense overrepresentation of the particular fragment that you're interested in. And one way to make it easier for you to figure out how much DNA is in here, because you can say, yeah, I know the volume of PCR product that I loaded in this well. I know the volume of how much ladder I loaded in this well, and the little product literature tells me that there'd be so much nanograms of DNA actually in this band here. I can then compare the brightness, right? And it would be made easier if I also thought ahead and not just ran one well with my sample, but maybe 2 or 3 wells, each one with a different dilution. Maybe the next one is 5 times less DNA. The next one afterwards is 10 times less DNA than my starting one. And then I would see different band intensities, and I could pick one that's maybe the closest in brightness to this ladder here. And that would give me an estimate of how much DNA that I have. You would then send that DNA off to be sequenced, right? And then the sequencing lab would give you the actual sequence of bases in this PCR product that you made. And then you can look it up and see if that actually corresponds to a particular species of, let's say, fly, right, that you're trying to identify. Okay, so those of you doing lab, you're going to be doing that. So hopefully that helps you understand a bit better conceptually and gets you excited about what you're doing. Right? So let's shift gears a little bit and talk about, now that we've been learning about DNA and how it's packaged and the idea that DNA contains information, right? Let's build on that. Let's get a better understanding of the significance of that information. So DNA contains stretches of information that we call genes. Right? So genes encode for RNA molecules that serve some sort of function. One way to think about it is if we have a long strand of DNA, for example, like this is a, I went to Long's and I bought something. I think I bought like 2 things, and then the extra coupons that come out are like, a yard long. I mean, you know how this is. But, so when you look at something like this, right, let's pretend this is a molecule of DNA. Maybe it's 1 chromosome, right? And maybe a genome has multiple chromosomes, right, on it. So the chromosome, if you read the information on here, right, it's just letters, numbers, punctuation, right? But we can make sense of it because we can read it, right? So there's a coupon for Revlon Cosmetics, one for Maybelline, Garnier facial skincare. I don't really care about those, right? Here's one for Gold Emblem Nut Mix, Trail Mix, or Salty Snacks. All right, okay. So if you were not literate, if you could not read at all, right, this would be meaningless. You would see that it's a whole bunch of shapes and things, but you wouldn't be able to interpret it, right? Think about the strand of DNA. The DNA is just simply A's, T's, C's, and G's, right? It doesn't have all the letters of the alphabet plus numbers plus punctuations to make sense of. When we look at this, we can tell where one coupon starts and one ends because it has like a header, right? It has like some big text. It has a little description, right? Here's the trail mix, and it has like this little barcode. We can tell that this bit of information is contained in this stretch of the print on here. So if I was just interested in the nut mix, this is the part that I'm interested in, right? So think about if this were a big long string of A's and T's and C's and G's, right? That's all our cell has to work with. And yet along the chromosome are these discrete stretches of information. And those are the genes, right? Okay, so the central paradigm or central dogma of molecular cell biology, this is kind of a famous phrase, the central dogma, is that the way living things carry out their cellular functions is by starting with DNA over here, right, and information in the DNA being transcribed, right, or converted into a different molecule, RNA, and then the information in that RNA is converted into a finished molecule, the protein. Right? And as we've already been talking about, proteins are long chains of amino acids. They're an example of one of these macromolecules. Right? So proteins, we said, are an example of a macromolecule that when it's folded up, it can perform different functions like little machines. So the essence of how our cell works is, right, our cell is filled with the small building blocks of these large macromolecules, free-floating sugars like glucose, right, free-floating fatty acid chains, right, free-floating nucleotides that can be then chained together to make nucleic acids, and free-floating amino acids that can be put together to make proteins. So what we're getting at here is one of the key central parts of this is the production of proteins, because the production of proteins, those are the machines that then can take the other free-floating types of monomers, like sugars, like a fatty acid, it's not really a monomer, but a fatty acid, or like a nucleotide, and put those types of polymers together, right? It's the proteins that put together the other types of macromolecules. So it makes sense then that studying this pathway of information is critical for understanding or appreciating how the cell carries out its functions. So we have somewhere on the order of 20,000 to 30,000 genes, right? So you might say, like, how come we're so bad at estimating how many genes we have? Well, again, because if I had a string of coupons and I said, how many coupons do we have here? You could very easily count how many coupons, because again, you know how to recognize one coupon from another coupon from another coupon. But if this were just a long string of A, T, C's, and G's, how can you tell what's actually a gene and what's not a gene, right? It's hard. So you can only identify them based upon the consequences of those regions of the DNA being actually used. Many genes are non-coding. They don't end up producing a protein, or the cell does not actually use them to make a finished protein. They just make some kind of RNA. Right? And the RNA molecules these certain types of small RNA molecules can serve different important functions just being a small RNA molecule, and we will study those. The way this cartoon is drawn, you can see the flow of information, the central paradigm or central dogma, starts from DNA in the middle, this dotted line, and goes to the right. Okay. Of course, our cells also utilize DNA when we have to replicate or copy the DNA for cell division. But that, you see, is a different process. That goes to the left. And it's important to keep in mind that a cell at any given time may be dividing, so going to the left here and doing something with DNA to replicate it, or carrying out its cell type specific functions. So in other words, going to the right of this figure, but it cannot be doing both at the same time. That's because the way the DNA molecule is being manipulated or accessed to perform these different functions, those functions are not compatible with each other. You can't do both at the same time. If I want to photocopy this, and make a perfect copy, like replicating my DNA, and I go to the photocopy machine, right, and I put it in the photocopy machine and close it, the photocopy machine is dumb, right? It doesn't understand anything that's on here. It's just making a perfect copy. But while that's happening, I cannot read it because this thing is in the photocopy machine. So therefore, I cannot interpret any of this information, right? So a cell, likewise, can go one way or the other at any given time, but it can't do both. And that's important when we talk about the cell cycle later on. All right. So remember, chromosomes are condensed as part of replicating, right? This is just a review slide. The DNA is packed up very tightly to make chromatin. So chromatin, again, is the DNA that's sort of organized in such a way to do things, such as make a long strand of DNA much more compact, so it's easier to manage. So now I can carry this around in my pocket. More importantly, when the DNA is tightly condensed, right, like this, or like the rods that you see on the screen here, this makes it much easier for the cell to be able to take 2 copies of the same chromosome. And if it's condensed them, so this is now chromosome 1, right? And you have two copies of it now, right? This makes it a lot easier to carefully divide these up so that each of the daughter cells can get a copy of the chromosome, right? But that's not how our DNA and our chromosomes normally look when the cell is performing its functions, right? Because keep in mind, when this is all wadded up like this, right, this is not compatible for me to read the receipt either, right? The DNA is not accessible, right? That trail mix, salty nuts coupon, I can't see it, right? Normally then, if the cell is functioning and doing its normal processes, the DNA, the chromatin is more relaxed. So that's the slide here. So this is a wonderful image. This is a computer model, but it's based on real data. This is what our chromosomes really look like. If you have a cell, when a cell is normally doing its normal functions, rather than being the chromosomes being in tight little rods, right? They're kind of more relaxed. And you can see here, you end up with these different sort of neighborhoods or territories where different chromosomes, right, their genetic information can be accessed. We will look at this a little bit more when we talk about how the cell dynamically changes the chromatin structure. All right, so let's talk more about this central paradigm then, this idea of how the cell uses and accesses the information in the DNA. We said that the central paradigm is information flowing from DNA through RNA and eventually, eventually as functional proteins. I mentioned that there are some forms of DNA that just kind of exist as some forms of RNA that just function as little RNA molecules that do certain things. Right? So this brings us to the 3 different types of RNA that we have in our cells. Messenger RNA is the RNA that contains the full information of the protein that's going to be made from it, right? So the message that's being carried by the mRNA, right, ends up being converted into a finished protein. Transfer RNAs are short little RNAs that form a 3-dimensional shape, kind of cloverleaf shape, that help out by helping to build this finished protein out of the mRNA message. rRNAs, or ribosomal RNAs, are special RNAs that are part of this huge protein complex in our cells known as the ribosome. So a ribosome is another example of many protein units that are coming together that are sort of wound up with these little rRNA fragments to give the ribosome its correct shape. And then the tRNA will work with the ribosome. And together they will put together an amino acid sequence based upon the message of the mRNA. And that amino acid sequence that's put together will be your polypeptide chain that then folds up as a protein. Right? So there are 2 kind of stages in this picture in which information is somehow being converted to a different form. We say when DNA is being converted into RNA, that is the process of transcription. When mRNA is being read and converted into a chain of amino acids to make a protein, that is the process of translation. Right? So the directional flow of information goes through RNA. So let's get these 2 different concepts clear. Transcription is essentially making copies of the DNA-based sequence, but making those copies in the form of RNA. Right? That is like photocopying it. Right? Translation is interpreting that genetic sequence that's in the form of RNA now and converting that into an amino acid sequence. So you might say, well, I don't get it then. How come this seems kind of like a roundabout way to make a protein? Because if the genetic information is all contained in the DNA, how come our cells don't just make proteins directly from the DNA, right? Because then that would have the utmost kind of integrity. The genetic information would be most accurate there. You wouldn't be going through this intermediate process of relying upon copies of the DNA in the form of RNA, right? Well, I have sort of a little story here to help you make sense of this, all right? Because there's many evolutionary benefits of going through RNA, right? If you use mRNA as an intermediate, it allows you, very importantly, to try to keep the DNA, the original DNA, protected in the nucleus. Because after all, we want to minimize the potential damage to our DNA. Because if our DNA gets damaged and our genetic sequence gets altered, right, we're in trouble. So by using RNA, you have an intermediate molecule that's a little bit more expendable, right? Because you can always make more mRNA from the DNA. Right? And the other benefit is that if you have a lot of RNA molecules made, a lot of mRNA, right, they can all be translated into the finished protein product at once. And therefore, you can make a lot of proteins by having a lot of mRNA floating around. And the third thing is you can edit the mRNA to make alternate proteins. Right? Okay, I'm being mindful of the time here. So I kind of have an analogy here to help you understand this, right? So, you know, I did my undergrad at UC Berkeley, and that's where I met my future wife, right? So we met as students at Berkeley, and when we were dating, you know, one of the places we like to go is right next to campus. There was this Mrs. Fields cookie shop. So this is in the early 90s, okay? And Mrs. Fields, you know, they had a nice little cookie store there, and we used to like buying cookies and things like that. And one day she decided to buy the Mrs. Fields cookie book from the store, right? So this is a very popular cookie book now, right? I think it's been around for a long time, okay? And so to this day, you know, my wife is a wonderful baker, so she likes to make cookies from this book, right? Let's pretend, right, that you are part of a club on campus or something like that, right? And as part of a club function, you need to make something like, you know, 100 batches of cookies, right? Let's treat this as the genome, because after all, right, this is a volume that contains information. So just like the individual coupons on my Long's receipt were individual packets of information, we can think of this also, right? In this case, each packet of information is a different cookie recipe, right? So the most famous one probably right at the beginning of the cookbook is Mrs. Fields, what is this? The blue ribbon chocolate chip cookie recipe, right? So it contains all the information here to make these wonderful cookies. So let's say that's what you want to do with your club. You want to make like 100 batches of these cookies. So you're not going to do it yourself, right? Because your kitchen in your apartment, you can't make 100 batches of cookies in time. So what do? You recruit several friends, right, who all have kitchens and who are all good bakers, and you say, we're going to follow this recipe. You could just pass this cookie book around, you know, your 12 friends or something like that, right? But then what happens? That slows things down. That's not efficient. Right? If you have to take turns using this. Also, there's the risk of this being damaged, right? You're going to pass this to 12 different kitchens. You're going to get flour or eggs or stuff poured on it. You might damage the original recipe. And in this case, this book is kind of sentimental, right? So what you do instead is you might want to first, instead of relying upon the original copy that's in the book, maybe you make photocopies, right? Photocopies of the recipe that are essentially identical to it. And then you can pass out these photocopies to each of your friends, and they can then take these photocopies and do something with it, right? They can go to their kitchens and you can make many batches of cookies at once based upon it. So the photocopy is the transcribed version of the original DNA sequence, right? So this is the mRNA molecule that is the perfect copy, or the RNA molecule, that's the perfect copy, right? But now you have many copies of it, right? So that one, you protect the original genome from being damaged. So this can go back onto your shelf, right, where it stays nice and safe. These, you don't worry if these get damaged because they're just copies, right? Two, you can make many batches of cookies at once because your friends each have a copy of this. They can now make it in their kitchen. So you can make 12 or more batches at once. And the third thing is interesting. Your friends might decide to edit this, right? So one of them might say, well, I don't like milk chocolate chips. I like dark chocolate, right? So as a result, they may scratch out the part that says milk chocolate and write in dark chocolate. Or another one of your friends, might say, I don't want to use sugar, right? I want to use Splenda. Their cookies are not going to taste as good, but I want to use Splenda. So they cross out the sugar part and put Splenda in there, right? That means their cookies that come out are going to be slightly different. They're going to be a slight variation of the original recipe, right? And that's a benefit because maybe one of their variations actually tastes better than the original recipe. And the important thing is, the original recipe, though, is not altered, right? So having an mRNA intermediate allows you to do those things: protect the original DNA, make a lot of cookies, or in this case, proteins, from that one gene originally, and you can make different versions of the protein. Okay, let's go back to the share here. Which is the one that I'm actually looking at? I get very confused here. Okay, so that's the analogy we're talking about. Hopefully you guys still see this. So it says a cookie or cookie analogy, right? Can one of you just unmute yourself and tell me that you see this?
Yeah, I can see it.
Okay, good, thank you. Yeah. So what we're saying is the original DNA, right, in the genome is the original cookbook. Transcription then is photocopying that to make this intermediate molecule, mRNA, right? And that mRNA can then be edited, whatever, and then that will be translated. So the fact that your friends have a copy of that and they go to their kitchens, they still have to do the work of actually interpreting that information and turning it into the finished product, right, which is the protein, the cookie. And hopefully you realize, right, there's a big difference here. The original information, like the photos of the cookies in the recipe, is not the same as the finished cookie itself, right? Yes, right, I actually have a Mrs. Fields cookie here, right? So I should open this. As though none of you guys have seen a cookie before in real life, right? This is really just an excuse for me to be able to eat a cookie at like 10:15 in the morning, right? So the important concept here is the finished molecule, the protein, right, is quite a bit different than the original information that codes for it, because this is what you want at the end of the day, right? The finished product of this central dogma of information flow is the finished protein molecule that's encoded for by that gene. Right, now I have crumbs all over here. I'm going to get roaches in my office, right? So again, baking cookies this way provides higher output, it provides flexibility, and it protects the original cookbook page from damage. That's why life evolved on Earth to use this RNA intermediate. Right? So different forms of life do this process differently. Right. Prokaryotes, the simplest ancient forms of life, they do sort of a coupled transcription and translation. In other words, as they are transcribing their genes from their genome into RNA, at the same time that RNA is being translated into polypeptide chains. So these light blue things are ribosomes, those protein complexes that are made of special proteins, ribosomal RNAs, right? And with the help of tRNAs that we will learn about later, these ribosomes are making a chain of amino acids coming out here by following the genetic code of the mRNA message that codes for what the next amino acid should be in this chain, right? And then this chain will come off, and this chain will then fold up as your finished cookie, right? Or or as your finished protein. Eukaryotes, like in our cells, we've evolved to separate the process of transcription from translation. So in our cells, transcription is done in the nucleus. So stretches of our DNA are transcribed. They're first made as a perfect photocopy, which we call the pre-mRNA or the primary RNA transcript, right? They can be edited. Right, so they can have different parts of it edited, just like your friends could edit the copies of the recipe they gave them to put in dark chocolate chips or Splenda instead of sugar. Right? That mRNA is the final edited version that then gets exported from the nucleus into the cytoplasm, where we also use, of course, ribosomes to then translate that message and make the polypeptide chain that original gene codes for, right? But importantly, the finished polypeptide, right, is a very different molecule than the original gene that encodes for it. Just like the original cookie recipe on the page of a book is very different than the finished cookie that you can hold in your hand and actually eat. Right? Oh my God, crumbs all over my desk. All right, so let's finish up by just talking about transcription then, this first step. So transcription, or the copying of the DNA, is done by another polymerase type of protein. Remember, polymerase is just a general term. A protein that makes polymers based on something is a polymerase, right? But in this case, it's going to make RNA, right, by reading the DNA. So this is an RNA polymerase, and in our cells, it is RNA polymerase II. It is the photocopy machine. That's what this big blob is. Again, another big complex of proteins working together as a machine. It's going to read the DNA. So this stretch of DNA has a gene in it that needs to be read. It unravels the DNA, and you can see it unravels it so that it can read, right? The strand of the DNA that provides the sequence in which it can then, the RNA Pol II puts together nucleotides, right, to make a string of RNA, and then the RNA comes off, right? So it reads this template strand, right? And this illustration down here introduces a couple of key terms. Here's the double-stranded DNA. One strand is what we call the coding strand. The one that's complementary to it is the template strand. Okay? It is, you can see here, the template strand is the one that the RNA polymerase II is actually reading, right? So in the cartoon up at the top here, that's this blue strand of DNA, right? That's the template. And you can see wherever there's a T, the RNA polymerase will now add an A in the growing strand of RNA, right? Wherever there's an A, it adds a U, because remember, this is RNA, so it utilizes uracil instead of a T, all right? And so on and so forth. We call the top strand the coding strand. This is a little confusing because conveniently, if we were to say, what is the sequence of RNA that comes off of this stretch of DNA, right? It's more convenient to refer to the coding strand, right? Because that is the one that at the end, right, has more, the letters are more similar, right? Okay. So here you can see that transcription starts at particular sites surrounding genes. These gold blobs are the RNA Pol IIs, right? They're going to attach at certain regions that we call promoters. Promoters have certain sequences in them that attract the RNA Pol II to start the process of transcription, right? So along one gene, you would have multiple spots where different RNA Pol II complexes are beginning at the same time to read different genes. In those promoter stretches of the genes, you'd find certain DNA sequences that are important for recruiting different proteins to be able to build up the complex that's going to get RNA Pol II to stick there and start moving. So these are the four main types of sequences that you find around a promoter. Right? This arrow indicates this is where transcription will actually start, where the RNA Pol II will start making an RNA sequence based upon the DNA sequence that goes off to the right here. But these other colored boxes, right, like TATA box and the BRE box, this is where other proteins are attracted and they bind. certain proteins bind to DNA to help the process of transcription. So we call them TFs, or transcription factors. So here's just some examples in this cartoon here. You have like a TATA binding protein, right? This protein that's represented as an orange ball. So it would bind to the TATA box sequence. And you can see here it is bound here. Then you have other proteins that also pile on, and eventually they create sort of a shape that is the right shape for the RNA Pol II complex to then also fit on there. And importantly, it's exactly the right shape in exactly the right spot so that the RNA Pol II is lined up so that when it starts transcribing at the intended spot. Okay, so I know I'm over time, so I apologize for that. But I wanted to finish with this little thing because I think you'll find this interesting, right? As you know, many mushrooms are poisonous. One of the most poisonous types of mushrooms is this Amanita mushroom, or the death cap mushroom. It's very brightly colored because, you know, it evolved to be brightly colored so that animals like deer, you know, they're smart, they stay away from it, right? We're the dumb ones. We're like, oh, what a pretty mushroom, I'm going to eat it. No, right? It evolved as a warning that it contains a poison, right? And this poison is these amanotoxins. All right, so here is an amanotoxin, ATX. It's the small little red molecule. This thing right here is the RNA Pol II complex that transcribes our genes. The amanotoxin is the right shape to fit perfectly right in the middle of where the RNA Pol II needs to be transcribing the DNA. And as a result, it stops RNA Pol II from transcribing your DNA. In other words, it prevents proteins from being made from the genes that a cell needs to be able to make, right? So in this case, it's a small molecule that we don't have in our body, right? But mushrooms make. And if we get this molecule in our body, it has disastrous results. That's why people die of mushroom poisoning, right? Because their cells in their body can't make proteins anymore. So I want you to think about this. The ATX it's just a small little three-dimensional molecule. There's nothing magical about it. It's not somehow sending out, right, death rays or something like. It's just the right shape to fit and get in the way of important cellular processes. And that's all a poison is. What is a poison? A poison is simply a molecule that doesn't belong in our body. And if it gets into our body, unfortunately, it's the right shape to mess up other molecules from working properly, right? And mushrooms, of course, evolved this as a way of self-defense. Okay. All right. So sorry for going over. I'm going to stop there. You know, this last slide is just like a little picture of showing you like a nice three-dimensional model of RNA Pol II going to work. Okay. All right. So let me stop the recording.